I'm sure most of you are aware that graduate school has a lot of challenges inherent in it. And I'm here to say that I think Mihaela has done a fantastic job of meeting and surpassing all those challenges. And that's an amazing thing to say, considering that for her they were stacked even higher than usual, I would say. She came from a very small college to a pretty big research-intensive university. That's a big adjustment. Came from the south to the north where we have blizzards, you know, when it's practically spring. That's a huge adjustment. She walked into a project that was very much at the formative stages and had a wonderful mentor at the beginning, but who didn't stay in the lab that long. So she was all of a sudden kind of almost on her own to shoulder a project with which she's done a fantastic job. And then, of course, graduate school is also about growing up. And here again, Mihaela came with the odds stacked a little more against her. She came quite young. She wasn't even old enough to vote, let alone drink when she got here. Well, legally. So there have been some rocky moments along the way. There have been some tough times and setbacks in the lab, but every single time she's come back and put everything into it and done a fantastic job with putting together what I think you'll agree is just a beautiful story. So, well done.
So hi everybody, thank you so much for coming today. I am terribly, terribly happy to see such a full room and see so many wonderful faces that I really care about so much, and I'm going to try not to get emotional so I can be professional. My mom is like, you know, be professional, show the Southern woman for what she is. So I'll try to give you a good story on that. Alright, so today I'm going to tell you about my story, which is understanding how insulin signaling in the fruit fly regulates a starvation response in the ovary. So, make sure my clicker works here. Hopefully it will. Alright, at any rate, so in the Drosophila ovary, you actually, many of you don't know what the Drosophila ovary looks like, so here's two ovaries here. And so each of these ovaries is actually made up of 16 assembly lines of growing egg chambers. So this is one of these assembly lines, and that's called the ovariole. Now at the very anterior tip here is the germarium, where the germline stem cells reside. And at the posterior here you go there, is a very well-developed egg that's about to be laid. Now, at the very tip of this, in the germarium, there's an interesting process that occurs that's actually common to a lot of developing germline cells. So, as you can see here, one of these germline cyst daughter cells actually undergoes a incomplete cytokinesis 4 times to yield a cyst of 16 interconnected cells, the germline cells. Now, one of these cells actually goes on to become the oocyte, or the egg cell, while the other 15 cells serve as nurse cells. And they actually provide the egg cell with things like protein, RNA, and organelles at specific times while that egg cell grows. Now, the way that a lot of these different factors, like RNAs and proteins, become enriched in that egg cell or the oocyte is by moving in large cargo units along these train tracks called microtubules. I'll try turning off, it'll take a second. So essentially, these large cargo units, there are many different kinds, but in this talk I'm going to be using the Oscar RNP, or ribonuclear protein complex. So what this includes is the Oscar RNA, as shown here, which becomes enriched in the oocyte, and many different proteins, and I've labeled 3 proteins that are included in this talk to mark this aggregate, that keep this RNA translationally repressed until it reaches the oocyte, where it can be de-repressed. Now, thank you. Now, this complex actually moves along these microtubules by this motor protein called dynein, which walks along microtubules towards the myosinus. Now, this works to help enrich various factors in the oocyte as these microtubules are nucleated in the egg cell and extend through these openings in the cytoplasm called ring canals. Now, because they're nucleated there, the myosinus are concentrated in the oocytes and the plusins are concentrated in the nerve cells, allowing for the directed movement towards the oocyte. And just to show you a quick movie here, you can see in the top, in this looped movie, one of these actual aggregates moving along microtubule towards that oocyte. Now, one other thing that's really neat about the Drosophila ovary is that it's actually exquisitely sensitive to nutrition. So, as I told you here, this egg chamber develops from that germline cyst cell and divisions there, all the way to this developed egg cell, or this developed egg. But in the middle, you have the telegenesis, or yolk uptake. So this process is where the fruit fly liver equivalent, called the fat body, actually secretes yolk protein that is taken up by this egg chamber. Oh, sorry. Now, under starvation conditions, the mother actually stops this the mother actually does not make this huge contribution of the yolk protein to the egg chamber, as in starvation conditions, these egg chambers actually go through this cell death, or apoptosis, which occurs about partway through the development at stage 8 or 9. Now this yields an ovary that's very small and looks tiny, but these earlier cysts look actually pretty normal until they reach that degeneration. So one of the things we were wondering in my project with Yuko Shimada was if these earlier stage chambers are actually sensitive to nutrient availability. What's going on in these earlier stages? Now, what Yuko did notice, and we published in our paper a couple of years ago, was that we actually do see a very unique and quickly reversible, as I'll show you in the next slide, response to starvation. So as you can see here, this is YPS-RFP, which is marking the Oscar RNP I showed you earlier. And you can see that it's highly enriched in this egg cell or oocyte, while it's pretty diffuse through the nerve cells. Additionally, tubulin is also pretty enriched in the egg cell, and then fairly diffuse, allowing for that transport process to occur. Now, with 24 hours of starvation, what you see is these RNPs actually form these large aggregates in the cytoplasm of the nerve cells, and the microtubules actually significantly condense to the cortex or sides of these nerve cells. So this is interrupting sort of that transport process we were seeing. Additionally, this starvation phenotype is quickly reversible. So as you see here, this is an egg chamber starved for 24 hours. Now within 24 hours of giving it normal food, so food supplemented with yeast, you can see that there's a massive reduction in the number of these aggregates in the cytoplasm of the nerve cells. Now, if you actually quantify this, you can see within 2 hours, you actually have a reduction of the number inside of these aggregates back down to normal levels when they're normally fed. So this is a very sensitive process that we're seeing here. So, to summarize the introductions, the salient points are that our fruit fly egg chambers are a great system to study polarized transport. And polarized transport is really, really neat for many reasons, but one of which is that that system of polarized movement I showed you is actually also in your nerve cells. So it's pretty neat that we can pull out fruit fly ovaries and look at that. Second, there is a cellular stress response before that apoptotic degeneration, and it seems to be involved in the polarized transport mechanisms. And finally, this response is exquisitely sensitive to nutrition. So the main questions in my project was, first, what exactly are these germline aggregates? What are they made of and what are they doing? Second, how are those external conditions being related to the ovary? How does the ovary know that the fly does not have enough nutrients? And finally, what is the significance of this starvation response? Is it actually important for the fly? So to start off with the germline aggregates here, I first looked through the literature and learned about different stress-induced foci that occur in the cytoplasm of various organisms. So as you can see here are mammalian cultured cells, and you can see in control conditions you don't have that many aggregates. But when you cause oxidative stress by applying arsenite to the media, you can see an induction of these large protein aggregates. And I'll talk to you on the next slide a little bit more about the identity of these aggregates. Additionally, you can see here in these yeast cells in glucose-positive conditions, so they have plenty of nutrition. They don't have many of these aggregates in their cytoplasm, but when you deprive them of glucose for even 10 minutes, you can induce these large protein aggregates in the cytoplasm. So just to briefly go over what stress granules and P-bodies are, first, what I thought they might be was stress granules, or sites of RNA triage or storage. So basically, the way that these form is when a translating RNA, so this is covered in a lot of ribosomes, thus polysomes. Now, under stress conditions, there are various kinases in the cell, like, for example, GCN2, that senses deprived amino acids, that can phosphorylate EIF2-alpha and lead to a stalling of the small ribosome subunit to create this 48S ribosome complex. Now, these can be further aggregated by many different factors, including poly-A binding proteins, into these large aggregates, stationary aggregates in the cytoplasm called stress granules. Now, when the stress is alleviated, these RNAs can be reinitiated into translation. Another kind of related body that shares some factors with stress granules, but not all, is the processing body. So this occurs where naked RNAs, or RNAs that are no longer covered in polysomes, are actually aggregated together with various decapping and degeneration factors, and are thought of as consequential degradation. However, they also have storage properties, and some RNAs can be reinitiated into translation. So a couple important things to note in this, as I'm going to actually be trying to figure out if my bodies are identical or have share properties with either of these, is that first, these processing bodies have no ribosomes, and they also have their host of unique RNAs that do, proteins that do localize to them. Additionally, stress granules have stalled ribosomes, and they also have their unique set of proteins that do localize to them. So first what I did is co-localization studies to see if they shared identity. So as you can see here, to start off with, they actually do seem to share a lot of components with processing bodies. Now, as you see in this top row, this is decapping protein 1, which, decapping protein, and then Pac-Man, which is an exonuclease, so these are kind of RNA degeneration factors. And then this transition initiation factor 4E. And you can see that all of these co-localize very nicely with the aggregates of the Oscar RNPs that I'm seeing in the cytoplasm. Additionally, there are areas that are completely free of both the small subunit of ribosomes and the large subunit of ribosomes in starved cytoplasm. Now, these areas that actually have a dearth of these ribosomes are enriched for factors of the Oscar RNP, like ME31B, suggesting that these actually are ribosome-free areas and share identities, once again, with processing bodies rather than stress granules. Finally, these areas, these large aggregates, are enriched for RNAs. So as you can see here, this is Oscar, the RNA I was telling you about at the beginning of the talk. But additionally, bicoid and gherkin, which are two other RNAs that become highly enriched in the oocyte, do actually co-localize with these large aggregates in the cytoplasm, suggesting that this mechanism of aggregation under stress conditions of these RNAs is common across various RNAs that are sequestered to the oocyte. Now, a final experiment I did on the note of are these processing bodies is to use cyclohexamine. So this drug actually prevents the ribosomes from coming off of the RNAs, blocking the translation. And so that actually leads to a decrease in processing bodies. So as you see here, when I first start culturing an egg chamber without cyclohexamine, if it's starved, it has plenty of these aggregates, it looks about the same at time 60 in culture, so an hour long. Now, if you add cyclohexamine to the media, you can see a dramatic reduction in the number of these aggregates in the cytoplasm, suggesting that they do share identity with processing bodies. Now, on the other hand, when you look at stress granule markers, you can see that these actually do not seem to share common identity with stress granules. So as you see here, stolphin, which is an RNA binding protein, does form its own aggregates in the cytoplasm, but they are not in common with our YPS marker here. Additionally, when I stain for EIF2 alpha, the phosphorylated form, as I told you, this is critical to the formation of classic stress granules. It actually doesn't form any aggregates in the cytoplasm. So it may suggest that under starvation conditions, at least, you don't see any classic stress granules in this cell type. Now, one other thing I looked at was a couple other cytoplasmic aggregates that are known to occur in the cytoplasm of this organism. So as you can see here, first I looked at U-bodies. So this is an assembling spliceosome complex. And as you can see here, though they do form their own puncta that are next to these processing bodies and exclusively next to the processing bodies, they do not actually co-localize. And there are a few labs that are starting to work on understanding a little bit more about what these U-bodies are and what they're doing. So I think it'll be really interesting to see, read up on what the interaction is between the spliceosome and the processing body. But that's yet to be learned about. Also, when I look at lysotracker staining, so this is trying to see if there's autolysis or some sort of degeneration that's occurring before that big apoptotic event. You can see that though there are a few dots, there's not a lot of these, and they do not co-localize with our large aggregates in the cytoplasm, suggesting that they are distinct. So what I've seen here is essentially that it is what our aggregates are sensitive to cyclohexamine. They have no ribosomes, and they actually have a lot of these factors known to be included in processing bodies, but not those included in stress granules. So for the purpose of the talk, I'm going to call these aggregates that you see processing bodies. So to summarize that part, first, these Oscar RNP components localize to large foci in these nurse cells. Second, they are like processing bodies and not autolysosomes. And third, they actually include RNP-enriched RNAs, so they might have some sort of protective role for these RNAs, and I'll go into that a bit later. So, the second part of the talk is understanding how these external conditions are communicated to the ovary. How does the ovary know the fly is being starved? So, what I'm going to show you is a story that basically talks about how the brain is secreting these insulin-like peptides into the fly, and these are actually recognized by the somatic tissue layer that surrounds the cyst of the 16 cells called the follicle cells. These follicle cells then communicate to the germline itself, and in fed conditions, when there's plenty of signal, you have dispersed microtubules and these RNPs are nice and diffuse. Now, in starvation conditions, you have less of these insulin peptides being secreted, less of this signal activated in the follicle cells, and then you get this phenotype in the germline. So how do I know that? Well, first, what is insulin and mTOR signaling? Now, there's a whole lot of factors in here, and this is just a brief overview of them. So what I'm going to do is zoom in on a couple of these. So first, the important thing is there is the insulin-like peptide. There are actually 8 in flies. The 8th was recently discovered. And these bind the insulin receptor on the membrane of the cell. So this activates a long cascade of various factors within the cell. But importantly, downstream, it controls transcription through defoxo and translation by its control of mTOR signaling. Now, mTOR signaling actually senses amino acid conditions and then increases the rate of translation through S6 kinase and through phosphorylating 4E binding protein. Now, this is actually positively controlled by insulin receptor, as when insulin signaling is activated, it depresses the repressor of mTOR called TSC. So, to go forward with this, what I first did was I looked at a reporter for insulin signaling. So phosphoinositides, or PIPs, actually localize to the membrane, PIP2 and 3 do, in increased levels when insulin signaling is active. So there's been reporter constructs made that tag one of these pH domains to a GFP, and this localizes to the membrane more when there's more insulin signaling. So as you can see here, in fed conditions, you see plenty of this localization of the GFP to the membranes, both the follicle cells and the germline. In starvation conditions, you see much less of this localization at the same exposure levels. So it shows that insulin signaling is actually affected in these starvation conditions. So in order to look at this a little bit more, what I did first was to culture these egg chambers with various compounds. So one of these things I used was actually insulin, which activates the insulin mTOR signaling pathway. And I also used rapamycin to decrease the insulin mTOR signaling. So first with insulin, and this is another time culture experiment, so you have your YPS RFP with the GFP tubulin here. And this is at time 5 minutes, so when I just start the culture, and this is an hour later in culture. And you can see that the starved control look about the same over that hour. Now, if you add insulin to the culture media, what you can see is there's a dramatic reduction in both the number and size of the foci, or the P-bodies, and also that cortical condensation of the microtubules, suggesting that this is insulin sensitive. Interestingly, this is pretty cool. Doing the reverse by applying rapamycin actually instigates a starvation-like response. So as you see here, in the control on the top, this is a fed chamber. Looks about the same, maybe a little worse over the hour. Now when you add rapamycin to the media, even in 5 minutes you start to see a bit of a starvation response, but after an hour you see a lot of these P-bodies in the cytoplasm, and pretty dense cortical condensation of these microtubules. So our drug experiments are suggesting, once again, that the insulin and mTOR signaling activity seems to be controlling this response. So as you see here, what I did was to try and control insulin secretion kind of from the top down. So control the secretion of insulin from the actual fruit fly brain. There are insulin-producing cells in the fruit fly brain, and these are called IPCs. So what I did is I either decreased the DILPS secretion from these IPCs, or I increased them. So what this looks like a little bit more clearly is I used KIR. So this actually decreases neuronal activity in these, specifically in the IPCs, to decrease the amount of activity they had, and that does decrease the amount of systemic insulin available in the fly. In this case, when I have decreased systemic insulin, I expected that they would look starved whether they were fed or starved, that they would think that there wasn't enough food around because they had low insulin inside. I did the opposite using TRIP. So this is actually a temperature-sensitive channel, and if you have it above 26 degrees, what it does is it's active all the time. You have plenty of insulin in the fly, whether or not there's food around. Below 26 degrees, it responds to stress normally and can kind of starve as a control. And the idea is that if you have increased systemic insulin all the time, they don't respond to starvation appropriately, and they look fed regardless of nutrition status. So, what I looked at here is you can see in the left when I do here, first the control, you can see in starved condition you have a lot of these P-bodies, you don't in fed. And when you express here, awesomely, you can see that you have P-bodies even when there's food around. So they think they're starving even when there's food. And once again, you see the reverse when you do TRIP. So here's TRIP at 18 degrees, so it's responding normally. You see plenty of punctum in the starved, but not in the fed. But if you express this TRIP above 28 degrees, you can see that it prevents them from mounting that normal stress response. They don't form these P-bodies in response to stress. So that's great. We know that systemic insulin seems to be controlling this response. So is it happening through that somatic follicle cell layer I was telling you about? Or does it go straight to the germline itself? Which of these is controlling that germline response? So what I did was I either used RNAi or transgenes in either the follicle cell tissue or within the germline itself to try and understand which of these was controlling. So, changing the follicle cell, the signaling level in the follicle cells, I either overexpressed insulin receptor itself to increase the insulin and mTOR signaling, or I overexpressed TSC1, which decreased insulin and mTOR signaling. So, what you see here is that actually I saw, once again, this exquisite control by the follicle cells. So, in the control, once again, you see this starved has plenty of P-bodies, but when you overexpress TSC1 and decrease the signaling, you can start this starvation response, all these P-bodies forming, even when there's food around. Again, when you do the opposite, so you overexpress insulin receptor and therefore increase the insulin signaling in these follicle cells, you can see that they become resistant to starvation and don't mount these P-bodies in response. So this shows that the follicle cell layer is in fact does in fact seem to control this signaling I'm seeing. But how about the germline? Previous studies have shown that the germline growth is actually controlled by insulin signaling. So I want to understand maybe that there's a direct response here as well. So what I did here was to do RNAi, or knock down the amount of insulin receptor or TOR itself in the germline. So basically the idea would be that your brain would secrete the normal amount of the insulins, like peptides, but the germline would not respond properly, and you should see P-bodies even when they're well fed. Now, I didn't see that. First of all, you can look here in the control. You have an egg chamber from the very tip, the germarium, growing all the way through to a fairly well-developed egg chamber. And when you have insulin receptor RNAi, and this was reported in germline clones, but it's never been shown in a full RNAi, that you actually have repression of growth here. You can see that these egg chambers just kind of stall out partway through their development. So that's pretty remarkable in and of itself, though it's been seen. But if you zoom in a little bit, what you see here is in fed conditions, they look just like controls, a little bit shrunken, sure, but they do not form these P-bodies when they are well fed, suggesting that perhaps the germline isn't doing the same control that the follicle cells are. Additionally, when you look at TOR, you see a similar sort of thing. Now, the growth restriction isn't as severe as you saw with the insulin receptor RNAi, but you do see with the germline not expanding to fill the space of the follicle cells quite as well as you do in the controls. Once again, when you look a little bit closer, in fed conditions, they do not mount these processing bodies, suggesting once again that this germline control is maybe more growth sensitive and less of a P-body. Though the genetics is a little tricky, so there might be more to find out about that. So, we know that our hypothesis, at least in this case, was incorrect, that disturbing the insulin pathway within the germline did not instigate the germline starvation response. So, now that we know that the insulin-producing cells are actually signaling to these follicle cells to control that response, what's happening on a smaller level? What's happening within the germline itself? So when I looked through the literature, I saw a few interesting things about some of these motor proteins, like kinesin and dynein, which may be familiar to some of you in the audience. So first, kinesin RNAi, so this motor protein that goes to the plus end of the microtubules, actually causes the microtubules to gather in these culture cells, and actually looks a lot like what I'm seeing in the periphery or the cortex of my starved germlines. Additionally, the kinesin is needed for the dispersal of stress granules. So as I was telling you earlier, though processing bodies and stress granules are distinct, they do share many common properties. So it could be possible that kinesin is controlling that in my system. Conversely, dynein is actually needed for the formation of stress granules. So once again, that could play into mine. So the idea is, do these motors actually play a role in this germline starvation response that I'm seeing? Interestingly, they seem to. So as you see here, first I did RNAi against dynein heavy chain, so I decreased the dynein. And as you can see here, in the control condition, the dynein look fairly like starved. They have very few of these aggregates in the cytoplasm. I do seem to see that the RNAi is working, as they don't form a very distinct oocyte, unlike the control. And the microtubules are very diffuse through the cytoplasm, as you can see here, perhaps even more diffuse than the fed controls. Now, in starved conditions, this is pretty neat. You can see here that they do not form the P-bodies in response to starvation. Additionally, these microtubules are very diffuse in starvation conditions. They just do not condense to the cortex like we saw in our controls. So that's pretty neat, and it suggests that this dynein is actually needed for the formation of these aggregates we're seeing and the microtubule phenotype we're seeing. Conversely, when I knock down kinesin, you can see here that in fed conditions, they have plenty of P-bodies in the cytoplasm, and their microtubules are always cortically condensed, regardless of whether they have food or not. So it suggests that these kinesins are actually needed for the egg to look normal or fed, where they have nice diffuse RMPs and diffuse diffuse microtubule condensation. So to summarize that part, basically the insulin-producing cells secrete these insulin-like peptides that signal to the ovary and control the germline starvation response we're seeing. Second, the follicle cells seem to receive this DILP signal and then transmit that to the germline by an unknown mechanism at this time. And finally, kinesin is needed for this kind of fed state of the microtubules and RMPs, and dynein is needed for our germline starvation response. So finally, we want to know a little bit more about what is the function of this response. What is the significance of it? Does it mean anything, really? So that gets a little metaphysical. So essentially what I did was I used the KIR and the TRIP, the tools I was using earlier, to think about whether instigating a starvation response, even when they're fed, causes issues with egg laying or the hatching of these eggs, so the health of the progeny, the fecundity of the fly. Or if I did TRIP, so making it think it's fed, even when there's no food around, seeing how that affects the progeny. So first, I'll show you the KIR here. So once again, to remind you, these are the ones that have the starvation response, whether or not they have food around. And as you can see here, I initially hypothesized with the egg lay that I would see a decrease of the egg laying to the levels that you see in control starved, since they look like they're starved when you look at the P-bodies. Interestingly, I did not see that. I do see a decrease in the egg lay, but it's not to the degree that I saw in starved conditions, suggesting that there may be some additional factors, probably ecdysin from the literature that's known, controlling this egg lay decrease you see. Additionally, I guess I got that covered. So next, I'll talk about how it affects the egg hatching. So as you see here, the first point you can notice is in these controls and in the KIR, you see that starvation itself, though they lay fewer eggs, these eggs seem to hatch at the same rate as their well-fed siblings. So even though they go through this starvation process, they seem to be pretty healthy. Interestingly, when you do decrease the systemic insulin constitutively with KIR, you do get a decrease in your hatching rate. So it seems like having that constitutive issue does cause an issue with the progeny. So to look a little more, to look at the reverse response, you can see your TRIP here. So these are the ones that are starvation resistant. They don't form all these P-bodies in response to starvation. And so I hypothesized once again, conversely to the KIR, that you would see an increase of the egg lay in the TRIP all the way up to your fed levels because they look fed all the time. Interestingly, they have that dramatic decrease in egg lay under starvation that is very similar to the control, suggesting that once again there's another factor, once again probably ecdysin, controlling this decrease here. So this is the really interesting point with regards to if it has importance. As you see here, once again, starvation itself doesn't really seem to affect significantly the hatching ability of the progeny. But when you prevent the progeny while they're developing from mounting that germline starvation response and make them go under starvation, they actually don't survive as well as their well-fed brethren. So this response actually may seem to have a protective mechanism, as if you don't have the response, the progeny are not able to hatch as well. So, to summarize that part, basically insulin alone does not control egg laying. Starvation alone does not control egg hatching, because they're healthy regardless. And also, preventing egg chambers from mounting the germline starvation response actually causes legs to be less healthy and hatch less. So, to kind of show the general model of my story today, as you can see here, in fed conditions, the insulin-producing cells of the brain secrete insulin-like peptides, which signal to these follicle cells. These in turn signal to the germline, allowing for this nice dispersed microtubule and RNP phenotype. Upon starvation, less of this signal gets out, and then you have less of these signal in the follicle cells, and accumulate these large RNPs or P-bodies in the cytoplasm, as well as this cortical condensation of the microtubules. Now, within these actual egg chambers, what this looks like close up is in fed conditions, these kinesins are allowing microtubules to slide on each other, keeping them in their nice diffuse pattern throughout the cytoplasm. This allows the dynein to track down the microtubules and kind of sequester the RNPs towards the oocyte. Now, in starvation conditions, what appears to be happening is you have less of this kinesin activity sliding the microtubules along each other, and the cortical dynein that is in the, we suspect, in the nurse cells, seems to be trapping these microtubules, causing them to be less stable, and causing less of these microtubules to be in the midst of the cell, causing an aggregation of these RNPs into processing bodies, as they're no longer being able to trucked away towards the oocyte. So that's the science part of the talk. I have acknowledgements after this. Should I go ahead and pause for questions at this point?
Questions?
Yeah, I'll go ahead and take the science questions first, and then I'll get into that.
Yeah, this is kind of, I don't know if it's really related or really important, but I'm curious about when you have the pathway and you have like amino acids with a question mark going to the insulin pathway. What is that?
Well, it's thought that some of these actually have a different charge on them, and that's what's sensed by the TOR. It's a little bit more fuzzy. That's kind of the idea, though, at the time, is that it's the charge on these amino acids that's leading to the sensation.
I guess I was wondering, it seems really simple to just add amino acids if that's an established
yeah,
pathway.
well, and also there are many different amino acids that can activate various things. So there are actually a few other labs, like the Partridge lab and that sort of thing, that are studying which amino acids control various responses in the fly, like your lifespan or your cell growth and that sort of thing. So it is a very interesting point, and we actually had somebody briefing in the lab who was playing with that, but she's no longer in our lab at this time. So I think that would be a really interesting avenue to pursue.
So when you say that the starvation response is reversible, does that mean that, so if a stage 7 egg chamber develops these P-bodies and then you feed the fly again, that same egg chamber is rescued, or the ones that are less developed?
So we think so, because in that 2-hour time span, there's not enough time for the egg chamber to grow and like pop out of the fly. It actually takes about a week for it to get to stage 7 or so, and then it's another couple days for that egg to develop into a full egg from that point. After vitellogenesis, it's much quicker, but because it's such a tight time window with the 2 hours, we do think that that same egg chamber should be able to recover. So it does in culture, at least, too.
I guess so. Just related to the biological significance when you do the TRIP expression,
Sure.
the eggs that do hatch. Is there anything wrong with the progeny?
So that's actually
versus the fed progeny?
That's actually something I was going to think about playing with and see. I think it's really important to understand a little bit more about how healthy are these progeny, how is their fecundity affected. I mean, there are various starvation things shown now that maybe your grandchildren could be affected. So I don't know. I'm trying to read up a little bit more on that literature. But.
Actually, just along with this question, most of these aggregates you're seeing has a lot of important patterning components that seem to be sequestered.
Right.
So I'm wondering if in the embryo, patterning is appropriate. Is it able to then survive, get to adulthood?
Because they do seem to, because the starvation itself doesn't seem to affect that, I think it's the sequestering that's going on may be a little bit protective, but I don't think it's probably going to affect the patterning, at least starvation. Now, in the ones that I did mess up with TRIP basically, I think it would be interesting to do stains on the embryos to see how, gherkin and that sort of thing are being affected. So,
You talked about the surrounding cells, the
Yeah.
core cells. Is it known how they communicate with those cells? I think there's some tunnels, but
There's
also interesting.
That's actually a huge question that I have been trying to pry at for a long time, and it's very much a black box situation. I think it's an obvious, really interesting next step, but I think it's better to work kind of up from the dynein and kinesin because we understand a little bit more about how those are regulated. Now there are, as you mentioned, some gap junctions that do connect that somatic tissue that surrounds the germline cyst with the germline, but it's been a little bit difficult to do the genetics to knock those out without really obliterating the health of the egg chamber. So I think that's an avenue that could be worked on, but I think we're going to try and kind of go from backwards up instead of up down on that. Any other science? All right, I'll do my thank you things. I'm gonna not cry, I think. Are we good? All right. So first, kind of the science things. I have a slide from my lab in a second. So thank you guys. I really do want to thank my thesis committee very much for all your advice over the years, and I've learned a lot from all of you, and I really appreciate your time and everything, very much. I want to thank the cell biology and genetics departments for all of the helpful advice and listening to my talks over the years, all the technical things, all the happy hours, lab interaction hours. We don't want to get too happy here. I want to thank Carl. He has been always an open door whenever I needed to just scan something by him, no matter how serious or lighthearted. Lisa Cotty for helping with all the logistics and everything, getting things done. Beth for logistics and also just being there to chat when I'm working on my figures at the computer next to her. Chris and for all of her help with scheduling and whatnot. And also the participants and developments. That's such a fantastic forum to learn about science, to talk about science, and just to know a little bit more, to get a little bit more exposure than you would in your normal journal clubs and seminars. Also, the fly community is a really fantastic resource, and I am so blessed to work with flies because you go to these meetings and everybody's so willing to help you and send you their stocks and antibodies, and it's been really great to be a part of that for the last several years, and I've been really lucky that Lynn's funded me to go to fly meetings every year, and it's been a pretty penny, but I've learned so much, and met such great people. I also want to thank my lab, who are awesome, because you guys are great. Lynn got down here at our wiffle ball tournament when we beat the weather line. Yep. We had a very victorious day then, sunny day outside. So I want to thank Lynn for being a wonderful advisor, and you've always had your door open for me at any time with any question, no matter how crazy my idea was or how I was that day. I really appreciate everything, and I really appreciate how much you care. And so I want to thank also current members of the lab. Andy knows everything. He's like the encyclopedia of science. He's just like, and he's always been very happy to sit down whatever he's doing and let me run something by him. And Kathleen for everything. She's lab mom. She just gets things done. And always with that hint of sarcasm that I just love. Like she makes me laugh every day. I also want to thank Peter, who's actually about to get out a publication, hopefully in Science really soon. We're really excited for him. He's done great. And Caitlin and Ronit, the newer students in the lab, but they have just been a joy to have. Very happy, great people. Previous members like Salji, who worked down the hall, Molly, who you can see there, is just great. She's just hilarious. And everybody else who's been in the lab. And I would be remiss if I didn't mention Yuko here. Lynn talked about my advisor that I had who had to leave fairly soon after I got here, and she has always been a great resource and taught me from the ground up. As Lynn said, I came from a very small college, and Yuko, she took me very patiently through everything. you know, I knew how to hold a pipette, but that was almost it. So she totally took me through and got me there. And Kenzie K is like, he's almost 6, probably he's like 5 or 7 years old. He's grown into a very lovely young man. So yeah. At any rate, so I love you guys. I've got to say, my lab has been like a second family to me, and I'm really glad I've worked with you all. So I'd like to also thank my family. So as you can see, my mama is here. I look like her, so I'm funny to be there. But even from when I was a little girl with my lovely little pet, I love that pet. She always had me up with her cooking. She always had me working with her, teaching me everything she could about life. And she's just been, thank you, Mom. You've been just such a source of support and love. And my mom, she always told me to shoot for the moon, and she always, even when she was shaking in her boots with terror, she always kept a plain face and was like, "You can do it. You can do it, baby." And I was like, all right. So I couldn't be where I am without her. I'd also like to thank my little sister, who these pictures do not do justice of how like crazy awesome she is, because she's just this brilliant, wise artist, and I love her so much, and she has been a source of creativity and inspiration for me my whole life. So thank you, sis. Also, my Uncle Johnny here, when he was in grad school, actually, he was living with my parents briefly, so I got to interrupt his sleepless nights in grad school with my baby kicking and crying and all that. So he's been one of the inspirations for me to get my PhD, and I love him very much. Also, my Aunt Kim, who's just been such a kind, loving, supportive person in my life, and she's just such a light every time I talk to her. Also, this is just my puppy Angel. She passed a few years ago, but I love her so much. So she was very, very cute and smart. And finally, I'd like to thank all of you guys, my friends. Grad school, as Lynn said, can be up and down, but I have been blessed with the most incredible people here. I mean, I really love you guys. I'm very, very blessed. So thankful for all of your support and laughter and love, and it'll be okay, and it won't be okay, but you'll be fine anyway. I couldn't have asked for a better, more supportive place to go to grad school, and better, more supportive people to know. So thank you so much for being here and being in my life, and yeah.
So the drill in cell biology is now, there's a committee meeting that with Mihaela and her thesis committee, which I hope won't take very long. And then so shortly after, so in just a little while, so probably around 2 o'clock, we'll have back in this room a celebration for Mihaela. So you're all invited to hang out and help Mihaela celebrate finishing her thesis.